Welcome back friends. Welcome back again to one more other lecture session from this channel. So in this lecture session we will be studying regarding transformation of DNA into host and bioprocessing in detail. So here there are several methods in order to transfer the foreign DNA into the specific host organisms like E. coli. But here prior to the successful transformation of DNA into the specific host organism like E. coli, the specific E. coli, it should remain in competent form. So here, once when the specific E. coli, when it remains in competent form, the specific E. coli, it is capable to intake the foreign DNA from the external source. Is it clear? So let me write it on the board. There are several methods. In order to transfer the foreign DNA from the external source, into the host organism like E. coli. But here, prior to the successful transformation of DNA into the specific host organism like E. coli, the specific E. coli, it should remain in competent form. So here, once when the specific E. coli, when it remains in competent form, it is capable to allow the foreign DNA from the external source. And here, most commonly, the foreign DNA insertion, it successfully takes place in case of the vector DNA at a specific site known as selectable marker region. So here, selectable marker region, it is the specific site in case of the vector DNA where the successful insertion of foreign DNA takes place. So here, once when the foreign DNA, when it successfully inserts in case of the selectable marker region of the vector DNA, now here it leads to the formation of recombinant DNA. So here after the successful formation of recombinant DNA, now the specific recombinant DNA, it is allowed to insert into the specific E. coli host. So here after the successful insertion of the recombinant DNA into the specific E. coli host, now here the specific E. coli host which is in association with the recombinant vector, it is allowed to grow in case of the antibiotic containing medium. Is it clear? After the successful insertion of the specific recombinant vector, now the specific E. coli host which is in association with recombinant vector, it is allowed to grow in case of the antibiotic containing medium. So here, as we all know, selectable marker region, it is the specific region in case of the vector DNA which successfully encodes the antibiotic resistant gene. So here, once when the foreign DNA, when it successfully inserts in case of the particular selectable marker region of the vector DNA, now the specific recombinant vector containing E. coli host, it is allowed to grow in case of the antibiotic containing medium. So here, only the transformants, they are capable to survive in case of the antibiotic containing medium. But whereas non-transformants, they are unable to survive in case of the antibiotic containing medium. So here, the antibiotic resistant gene which successfully encodes in case of the selectable marker region, it acts as a selectable marker in order to differentiate the transformants from the non-transformants. Is it clear? So let me write it on the board. Most commonly, the successful insertion of the foreign DNA into the vector takes place at a specific region known as selectable marker region. Selectable marker region. So in general, the selectable marker region, it encodes antibiotic resistant gene. So here, once when we insert the foreign DNA into the selectable marker region, now here the specific vector, it has become recombinant vector. So here after the successful formation of recombinant vector, now the specific recombinant vector, it is allowed to insert into the specific E. coli host. Is it clear? After the successful insertion of the foreign DNA into the selectable marker region, which encodes antibiotic resistant genes. Now it has led to the formation of recombinant DNA. Now here the specific recombinant vector DNA, it is allowed to insert into a specific E. coli host. Is it clear? So here after the successful insertion of the recombinant vector DNA into the E. coli host, now here the specific E. coli host which is in association with the recombinant vector, it is allowed to grow in case of the antibiotic containing medium. So here only the transformants, they are capable to survive in case of the antibiotic containing medium, but whereas the non-transformants, they are unable to survive in case of the antibiotic containing medium. Is it clear? So let me write it here. After the formation of or after the insertion of the recombinant vector into the E. coli host, the specific E. coli host which is in association with recombinant vector. Is it clear? The specific E. coli host which is in association with recombinant vector allowed to grow in the antibiotic containing medium. So here only the transformants, they are capable to survive in case of the antibiotic containing medium, but whereas non-transformants, they are unable to survive in case of the antibiotic containing medium. So in this way, the specific antibiotic resistant gene which is most commonly encoded by the selectable marker region, it acts as a specific selectable marker in order to identify the transformants from the non-transformants. Is it clear? And then after we will be studying regarding obtaining the foreign gene product in detail. Is it clear? So let me erase the things here. As already mentioned, then after we will be studying regarding obtaining the foreign gene product in detail. So here, after the successful insertion of the foreign gene containing DNA into the specific vector, now here the specific vector, it has become recombinant vector. Is it clear? So let me show it here. So here once when we insert a foreign DNA or foreign gene containing DNA into the specific vector. Now here the specific vector, it has become recombinant vector. And here the specific recombinant vector, it is allowed to insert into a specific E. coli host. Now here, likewise the other genes of the vector DNA, even the foreign desirable gene, it also undergoes successful expression in order to regulate the production of one specific target recombinant protein under optimal conditions. Is it clear? So let me write it on the board. After the insertion of the foreign DNA, into the vector, the specific vector has become recombinant vector. And here, likewise the other genes of the vector DNA, even the foreign desirable gene, it also undergoes successful expression within the heterologous host in order to regulate the production of one specific target recombinant protein under optimal conditions. So let me write it here. Likewise, the other genes of the vector DNA, even the foreign desirable gene, also undergoes expression in the heterologous host, in order to regulate the production of target recombinant protein. So here most commonly the specific recombinant target protein which is produced in case of heterologous host, it is produced at smaller scale under optimal conditions. So the reason why after cloning the specific gene of interest, in order to regulate the production of the recombinant protein under laboratory conditions, here we can take the advantage of one more other alternative strategy in order to regulate the production of the specific target recombinant protein at larger scale. And here the specific strategy is nothing but growing the specific cells in case of the culture medium. So here once when we grow the specific biological cells in case of the culture medium, so here by taking the advantage of the specific nutrients which are available in case of the culture medium, the specific biological cells, they are capable to regulate the production of the recombinant proteins or the target proteins at larger scale. Is it clear? So let me write it on the board. Most commonly the target recombinant proteins, which are produced in heterologous host under optimal conditions, they yield the recombinant target protein. at smaller scale, under optimal conditions, Here, after the successful production of the recombinant target protein in case of heterologous host under optimal conditions, now here the specific desirable protein, it can be successfully extracted from the culture system by taking the advantage of various separation techniques like columnar chromatography and ion exchange chromatography. And even the specific biological cells, the number of the biological cells, it can be successfully manipulated by growing the specific biological cells in case of the continuous culture system. So here what is the uniqueness of continuous culture system? Here the uniqueness of the continuous culture system, here it is nothing but in case of the continuous culture system, a specific fresh medium is added continuously from one particular side, but whereas from the other side the existing medium, it is drained out continuously. So here because of the addition of the fresh medium from one particular side and removal of the existing medium from the other side, the specific biological cells, most commonly they remain metabolically active or physiologically active at log phase or exponential phase. Is it clear? So let me arrange the things here. So here, as already mentioned, after the successful production of the specific protein product in case of the specific culture system, now here the specific protein product, it can be successfully extracted from the culture system by taking the advantage of various separation techniques. And some of the separation techniques which are most commonly used in order to obtain the specific recombinant target protein from the culture system, they may include ion exchange chromatography and columnar chromatography. Is it clear? So let me write it here. The recombinant target protein can be successfully extracted from the culture system by taking the advantage of various separation techniques, and even in order to manipulate the number of the biological cells, here we can grow the specific biological cells in case of the continuous culture system. So here what is the uniqueness of the continuous culture system? Here once when we grow the specific biological cells in case of the continuous culture system, fresh medium is added continuously from one particular side, and whereas the existing medium, it can be drained out continuously from the other side. So here because of the addition of the fresh medium from one particular side and draining out the existing medium from the other side, so here most commonly the specific biological cells, they remain physiologically active in case of the log phase or exponential phase. Is it clear? The specific biological cells can be manipulated by growing the specific biological cells in continuous culture system. So here in case of the continuous culture system, fresh medium is added continuously from one particular side. In continuous culture system, fresh medium is added continuously from one particular site, is added continuously from one particular site, and whereas the existing medium is drained out continuously from the other side. So here, because of this unique nature, in case of the continuous culture system, most of the biological cells, they remain metabolically active or physiologically active in log phase or exponential phase. So here, by taking the advantage of the continuous culture system, here we can manipulate the number of the biological cells in a short period of time. But here we cannot increase the number of the bioproducts. So here, in order to increase the number of the bioproducts, here we need to grow the specific biological cells in case of a specific culture vessel known as bioreactor. So here, once when we grow the specific biological cells in case of one particular culture vessel known as bioreactor, by providing suitable conditions, here we can increase the number of the bioproducts. Is it clear? So let me write it here. By taking the advantage of continuous culture system, here we can manipulate the number of biological cells. And here, in order to increase the number of the bioproducts, here we need to grow the specific biological cells in case of one particular culture vessel known as bioreactor by providing suitable conditions. Is it clear? So let me arrange the things here. So here, as already mentioned, small volume culture systems, they cannot yield higher production of biological products. So here, in order to yield high production of biological products or bioproducts, here we need to take the advantage of one specific culture vessel known as bioreactor. So here, once when we grow the specific biological cells like plant cells, animal cells and microbial cells, in case of one particular culture vessel known as bioreactor, by taking the advantage of specific raw materials, the specific biological cells, they are capable to induce the production of bioproducts like antibiotics, enzymes and organic acids. Is it clear? So let me write it on the board. Small volume culture systems cannot yield high production of bioproducts. So here, in order to acquire high number of bioproducts, here we need to culture the specific biological cells in case of one particular culture vessel known as bioreactor. And here the capacity of the bioreactor, it may range around 100 liters to 1000 liters. Is it clear? In order to acquire high production of biological products, here we need to culture the specific biological cells, the specific biological cells in culture vessel known as bioreactor, culture vessel known as bioreactor. And here the capacity of the bioreactor, it may range around 100 liters to 1000 liters. And here within the specific culture vessel known as bioreactor, specific raw materials are subjected. So here by taking the advantage of specific raw materials, the specific biological cells, they are capable to yield the production of many of the bioproducts like antibiotics, organic acids and enzymes. So let me write it here. In order to regulate the production of many of the bioproducts in culture vessel like bioreactor, certain raw materials are subjected to the biological cells in order to yield the production of many of the bioproducts like organic acids, antibiotics and enzymes. To the biological cells in order to yield the production of many of the bioproducts like organic acids, antibiotics and enzymes. Organic acids, antibiotics and enzymes. And here, in order to regulate the production of the specific bioproducts in case of one particular culture vessel like bioreactor, here we need to provide specific suitable conditions like temperature, pH, oxygen. Is it clear? So let me write it here. The bioproducts can be produced in culture vessel by taking the advantage of certain suitable conditions like temperature, pH, substrate and oxygen. So here by taking the advantage of all these specific things, here the successful production of bioproducts they take place in case of one particular culture vessel like bioreactor. Is it clear? And here most commonly the specific bioreactors which are most commonly used in order to regulate the production of many of the bioreactors, they may include stirred bioreactor. Is it clear? Stirred bioreactor and the other one is sparged bioreactor. So let me arrange the things here. Here, as already mentioned, in order to regulate the production of many of the biological products in case of the specific culture vessel like bioreactor, here there is a requirement of many of the specific factors like temperature, pH, substrate, oxygen and vitamins. So here by taking the advantage of all these specific factors, successful production of bioproduct that take place in case of specific culture vessels like bioreactor. And here, if you look at the specific bioreactor which is most commonly used in order to regulate the production of bioproducts, so here the specific bioreactor, here it is nothing but simple stirred tank bioreactor. So here, in case of simple stirred tank bioreactor, there is an availability of one specific culture vessel which is having curved base. Is it clear? So let me write it on the board. The most commonly used bioreactor in order to regulate the production of bioproducts, so let me arrange the things here. Production of bioproducts in case of culture vessel, like bioreactor may include simple stirred tank bioreactor. So here most commonly the simple stirred tank bioreactor, it has a specific culture vessel which is having curved base. So here because of the presence of the specific curved base, all the specific contents they are capable to mix so accurately by taking the advantage of a specific system known as agitator system. Is it clear? So here agitator, here it is nothing but what? Agitator, it is the mechanical stirrer which is responsible for mixing all the specific contents in case of the specific culture vessel like bioreactor. And even the specific mechanical stirrer like agitator, even it is also responsible for mixing the specific oxygen throughout the bioreactor. Is it clear? Simple stirred tank bioreactor. Simple stirred tank bioreactor has culture vessel which has curved base, because of the presence of curved base, all the specific chemical constituents, are capable to mix accurately, and even the specific mechanical stirrer, even it is also responsible for mixing the specific oxygen throughout the bioreactor. So here because of the availability of oxygen throughout the specific bioreactor, here we can observe bubbles. Is it clear? So let me write it here. The specific mechanical stirrer, it is responsible for spreading the oxygen throughout the bioreactor. And even the simple stirred tank bioreactor, it has many number of controlling systems. So here some of the controlling systems which govern the specific simple stirred tank bioreactor, they may include temperature controlling system. Is it clear? Temperature controlling system, agitator controlling system, pH controlling system, and substrate controlling system. Is it clear? So let me write it on the board. Many of the controlling systems also govern the production of bioproducts in bioreactor may include temperature control system, temperature control system, agitator control system, pH control system, and even foam control system, and many number of sampling ports. Is it clear? Many number of sampling ports, they are also present within the specific simple stirred tank bioreactor. So here, because of the presence of all these controlling systems, here we can obtain small amount of bioproducts periodically from the specific bioreactor. Is it clear? And then after we will be studying regarding downstream processing in detail. So let me arrange the things here. And then after we will be studying regarding downstream processing. So here, what is downstream processing? Downstream processing, right? So here, after the successful production of many number of the bioproducts by taking the advantage of specific bioreactor through the biosynthetic stage, now here the specific bioproducts, they has to undergo downstream processing. So here in case of downstream processing, there is a requirement of many number of purification techniques and separation techniques. So here in case of downstream processing, most commonly the specific processing mechanism, it takes the advantage of many number of purification techniques and separation techniques. Is it clear? So let me write it on the board. After the production of bioproducts in the bioreactor through the biosynthetic stage, now the specific bioproducts has to undergo downstream processing. So here basically the downstream processing, it takes the advantage of many number of purification and separation techniques. Basically, the downstream processing takes the advantage of purification and separation techniques. separation techniques. And in addition to this, even the specific culture products which are most commonly produced in case of the bioreactor, they need to take the advantage of many number of preservatives. Is it clear? Even the specific system, even the specific formulated system which has successfully produced in case of the bioreactor, it need to take the advantage of many number of preservatives. And even many number of clinical trials they has to undergo if the specific bioproduct which has successfully produced, if the specific bioproduct is a clinical drug. Is it clear? The bioproducts which are produced takes the advantage of many number of preservatives. And in addition to this, even the specific system, it need to take the advantage of clinical trials. If the specific bioproduct which has successfully produced in case of the bioreactor, if it is a clinical drug. Is it clear? In addition to this, even clinical trials are required if the specific product which has obtained if it is a clinical drug. And in addition to this, even quality controlling system, even it is also to be monitored. Is it clear? Even strict controlling system and quality controlling system, even it is also be required in order to acquire the specific bioproducts within the bioreactor at accurate level. Strict control system and quality controlling system. Even it is also required in order to obtain the bioreactor, in order to obtain the specific bioproducts in the bioreactor at accurate level. Here, the quality controlling system and even the downstream processing, it remains vary from one particular product to the another product. So here, in case of all the specific products, the downstream processing and even the quality controlling system, it won't remain similar. It remains vary from one particular product to the another product. Is it clear? So here, this is regarding transformation of DNA into host and bioprocessing from biotechnology principles and processes. And in the next upcoming lecture session, we will be studying regarding biotechnology and its applications in detail. So here, I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.